biliverdin, cholesterol, drugs, etc.
Correct.
And then animals excrete one major type of waste, that's nitrogenous waste.
Yeah.
That is usually ammonia, urea, and then uric acid. Ammonia is the most toxic. Urea is moderately toxic. Uric acid is the least toxic. And then ammonia is usually in ammonotelic, and urea is in ureotelic. Uric
No,
acid
so
is in,
those organisms that excrete their nitrogenous waste in the form of ammonia are called ammonotelic. And because ammonia is most toxic, it also requires the most amount of water to dilute in before excreting it out of the body. Correct? Yeah, continue.
so example of ammonotelic is a fish, that's freshwater, and then ureotelic is a marine fish, mammals, amphibians, and some reptiles. Then in uricotelic, you have birds, insects, reptiles, and snails.
Yeah, where do you think humans come? Humans, where will you place humans? In which category, Jania? Are we ammonotelic, ureotelic, or uricotelic?
Ureotelic.
Yes, we are ureotelic. Correct. Yes, continue. Continue, Jania. You are mute if you are speaking. Am I audible to you, Jania? Okay, I think Jania cannot hear me. So, are you there, Yusuf? Am I audible, people, at all? Hello? Hello, am I audible? Iram, Devangi, Jania, am I audible?
Yes, sir.
Yeah, okay, great. Finally, I thought I'm not audible. Okay. Jania, am I audible to you? Would you like to continue?
Yes, sir. Actually, for the diagrams that you explained, I don't have the notes. I just like drew it in my notebook. So
Okay. Now, so just to explain that what part is what. So you have a aorta, inferior vena cava, that supplies the blood. Aorta brings the blood to the kidneys. Inferior vena cava takes it back to the heart. Then I told you that in a kidney, this is all from your NCRT only. Okay. So just have to read your NCRT to understand the anatomy. Okay.
Yes, sir.
Yeah. So I also ask you to write certain things. Did you note that? We
Yeah,
also
like
have Iram
some
joining.
points. Yeah.
Okay, cool. Can you hear me, Iram? Are you there?
Yes, sir.
Yes. So would you like to give a quick summary for human excretory system? These diagrams, basically. If you.
So I'm very sorry, I'm not at my place and I don't have the notes right now with me.
Okay. Okay. Cool. No issues. Okay. So these two diagrams were just explained, and I dictated you things which were important for your boards from your NCRT. Okay. So please go and read NCRT also, right? So in today's class, let me give you proper like info and notes regarding whatever we studied in these diagrams. Okay, so we studied certain names. So some of these names in human excretory system were like Bowman's capsule, proximal convoluted tubule, columns of the Bertini columns or columns of Bertini, right? These things I taught you in the last class. So let's just um, define these before we go to urine formation. Okay, so write down. Let's go one by one. What are these things? So write down. First is columns of Bertini. Write down. These are the renal columns. These are the renal columns. Renal means kidney. Renal column, or in bracket you can write columnar structures. Present between the medullary pyramids of kidneys. Okay, so if you see the diagram here, yes, this one. So these are the medullary pyramids, which let me show by blue. So this is one medullary pyramid, this one, and this is the other medullary pyramid. This is the third medullary pyramid, and in between, if you see this space, so this renal column, this space here, that is called the column of Bertini. So this here, this here, so these are the columnar structures. Make sense? Everyone? Now next is nephrons. Okay, great. Nephrons. So these are the structural and functional unit of kidneys. Mostly functional unit. Write down. These are the functional unit of kidneys that help in filtration of the blood. Produce urine. Okay. Then we have the Bowman's capsule. Okay, what do we mean by Bowman's capsule? If you see the structure, we talked about this part here, this capsular structure here, this part is the Bowman's capsule. Okay. Now write down, it is a double walled cup-like structure. Double walled cup-like structure. Which encloses the glomerulus. Simple. Which encloses the glomerulus. Okay. Now glomerulus we talked about, it is a very convoluted structure made by the blood vessels, like basically the blood capillaries, which is present in the center of the Bowman's capsule, right? I hope you remember these structures. This is glomerulus, right? You can see the cursor, everyone. This here. Yes, no. Okay. And then coming to PCT and DCT, the proximal convoluted tubule and the distal convoluted tubule is a tubular network of these. It's like pipelines from where the filtrate moves. And today I'll be teaching you when this filtrate moves, why there is blood vessels around it and what happens to this filtrate and all. Okay, so we'll talk about that. So is this basic thing clear? So PCT, DCT, we will talk about when we are studying the urine formation. So anyone has any questions, or should I now go to urine formation? Any questions, people?
No, sir.
No? Okay. So let's talk about urine formation. Okay, now the first thing is when we talk about urine formation, we have to understand two things. What kidneys filter as filtrate is not the urine. Okay, urine is the final volume that we excrete out from our bodies. Okay, is it clear, everyone?
Yes, sir.
Yeah. So what is the role of kidneys is to just filtrate out. And as I told you in previous classes, our kidneys knows just one job. That job is to filter anything from the blood that looks like a impurity in the blood. Okay, so apart from blood itself and the cells of the blood, kidneys have the potential to filter out almost everything. Okay, so the first step in this, so urine formation is a step, it's a stepwise manner process. So first step is called glomerular filtration. Okay, which means filtration of blood. Glomerular filtration. Okay, so write down. This means filtration of blood and on an average, per minute, kidneys can filter around 1 to 1.2 liters of blood per minute. Okay. Now we know that what is the total amount of blood that we have in our body, our average human has in the body?
5 liter.
Yes, around 5 liters, right? At max, like males have 5 to 5.5 liters. Females may have, just because of the lower musculature and body mass, little less blood. Okay. But if kidneys can filter around 1.2 liters of blood, then how much time do you think kidneys will take to just filter all our blood out? Just a few minutes, right? Then why does kidney, so does kidney just works for 5 minutes every day? No, right? It's constantly filtering out. So what it is filtering out constantly? So it is filtering out almost everything from the plasma except for the proteins that cannot pass through the Bowman's capsule. Okay, so this filtration takes into consideration that very fine particles will pass through Bowman's capsule and will get filtered, and bigger particles will stay stuck in the Bowman's capsule, like your RBCs, WBCs, bigger proteins. So all the blood cells cannot pass. Also bigger proteins or proteins cannot pass. Okay, so this process is called ultrafiltration. So these, write down, the epithelial cells of Bowman's capsule have filtration slits. These are spaces which are present between the cells. So if there are two cells of this Bowman capsule, you will see that there will be slits like this. Okay. Can you see that I'm making small slits between two cells? So these regions are called slits, through which filtration happens. Okay. And these cells of kidneys are called podocytes. Is that clear, everyone? And write down, through ultrafiltration, almost all the components of the blood plasma through ultrafiltration, almost all the components of the blood plasma passes through the Bowman's capsule, passes through the Bowman's capsule except for the proteins. Proteins don't pass through the Bowman's capsule. Also write down the blood cells or the blood corpuscles, RBCs, WBCs, and platelets. They also do not pass through the Bowman's capsule. Do not pass through Bowman's capsule. Okay. And that's why it's called ultrafiltration. Okay. It's called the process of ultrafiltration. And whatever comes out of it is called the filtrate. So whatever is filtered is called filtrate. Now this filtrate is not urine. Urine is a waste substance excreted out. This filtrate is just whatever is not blood or the protein of the blood filtered by the Bowman's capsule. Now you can imagine a lot of useful substances also come through this filtrate, right? Come into this as a part of this filtrate, correct? Agree? Yes, no? Okay. So this filtrate, which is produced through the glomerular or Bowman's capsule, it is formed at a rate. So per minute, there is lots of filtrate formed, but all of it does not go on to form urine. So to make this filtrate, we have to understand another concept called GFR, glomerular filtration rate. Glomerular filtration rate. Okay, GFR. Now write down what is this? It is the amount of filtrate formed by the kidneys, both the kidneys, okay? Formed by both the kidneys per minute. Per minute. So I'm just talking about the amount that, so remember that efferent arteriole brought the blood in, that blood vessel then formed a glomerulus, and then the efferent arteriole took the blood out. And this was under this kind of a structure called Bowman's capsule, right? This is Bowman's capsule, and this is glomerulus. So this filtrate is just whatever amount passes from this into this, from the glomerulus into the Bowman's capsule. Is it clear, everyone? Okay. Now this filtrate is too much. So this rate is around 125 GFR is equal to approximately 125 milliliters per minute. That's very huge amount. If you will solve this, it will come out to be in a day, in a 24 hours, it will be around 180 liters per 24 hours. This is the amount of filtration that our kidneys do every day. We just have 5 liters of blood, out of which the filtrate, so basically it's the same blood that keeps on getting filtered, hundreds of times. Every minute we form 125 ml of filtrate. Now clearly we cannot afford to lose all of this, right? Correct? And also kidneys do have their own mechanism to regulate this filtration rate. This filtration rate is not fixed. It can go up and go down depending on conditions and situations. But the fact is that it is too much. It is like you cannot afford to lose. You cannot actually lose 180 liters from your body. You don't have 180 liters in your body. First point. But this amount of water or filtrate which is produced is clearly not a waste substance. So what happens is the second step. After this, when glomerulus has filtered the GFR, the next thing that happens is called reabsorption, which is a very, very important component, reabsorption. And this reabsorption happens in that PCT, loop of Henle, and the DCT. Okay, is it clear? So if you see the conversion rate, that's where this reabsorption comes. Because of this reabsorption, this 180 liters of filtrate, finally, when the urine formation happens, only 1.5 liters approximately of urine, 1.5 liters of urine is produced per day. So rest around 178.5 liters gets reabsorbed back per day. So it's more reabsorption happening. So it's more than 99%. So it's more reabsorption happening than filtration. So the kidney cells are evolved to do both reabsorption majorly and filtration also. Now let's see what happens. So for that, let's come to the functions. So is this part clear? Reabsorption and filtration. Okay. Okay. So now let me put this on image. Yes. Can you all see this image? Okay, let me make it a little big. Is it now clear? You can see it better this way. Okay, now look at this image and try to understand. So it's just a simple diagrammatic representation of a nephron without the blood vessels. Okay, so let me use blue here, or yeah. So you can see in the kidney, this is the cortical part, cortex of the kidney, this shaded area, and then this is the medulla of the kidney, where the medullary pyramids is. Okay, two layers. And this nephron spans from cortex to medulla. Okay, so these kind of nephrons which start from the cortex also go into the medulla are more in the kidney, and they are the most functional ones as well. So you can see that this part here is glomerulus, correct? This is glomerulus. What is this part? The Bowman's capsule. Then from here to here, just one moment. So from here is your PCT, proximal convoluted tubule. Then this is your distal convoluted tubule. And then you have your loop of Henle, descending loop of Henle and ascending loop of Henle. So let's first talk about just the reabsorption and secretion that happens in the nephron. Then we will add the blood vessels to it and we will see the complexity. So first thing is what is reabsorbed or secreted from the PCT. So let's write about PCT first, write down. The PCT or proximal convoluted tubule has cuboidal epithelial cells. Okay, cuboidal epithelial cells look something like this if I draw them. So, and they have brush-like borders, okay, to increase the surface area. They look something like this. They are attached to a substratum. They have a nucleus. And so these are cuboidal. They have a Golgi apparatus and their own mitochondria, etc. So this is a cuboidal epithelium cell. It looks like in 3D, it's like a cuboid. Okay. Epithelial cells of PCT. Okay. It's also known as the brush border epithelium. It's present here in this PCT. Now, what is the function, do you think, of these brush-like structures? What is the function of this filamentous? Similar kind of thing you see in your intestines as well. They are called villi. What is the function?
They increase the surface area.
Perfect. So here also you need to reabsorb things. So whenever there are functions in biology, one thing is the is the rule of thumb. So the thumb rule is that various structures evolve to help with the functions. Wherever the function is absorption, you will see that the cells have villi-like or finger-like projections, or these brush border-like appearances, cilia, etc. So they increase the effective surface area for absorption. Here also reabsorption is happening. So whatever came into this tubule here from the glomerulus, it has lot of useful substances in it. So what will happen? Lot of useful substance will be reabsorbed back by the cells. Okay, so write down. This increases the surface area for reabsorption. And PCT is lined by these kind of cells. This increases surface area of reabsorption. And you will see that PCT is the major reabsorbing component. Okay, so everything that Bowman capsule took from the glomerulus, from the blood, around 70 to 80% of that is reabsorbed back just in the PCT. Because PCT says that, see, I understand that you have got just one job, and that is to filter out things. But see, you are filtering out too many important things. You cannot keep filtering at this rate. And even if you keep filtering, see, I have to bring things back. Okay, so there is a negotiation, there is a cause and consequence effect. And around 80% of substances, which includes electrolytes, which is the iron and water majorly, is reabsorbed back in this segment. So you can see here what all things are reabsorbed. HCO3, bicarbonate ion, NaCl, which maintains the electrolyte balance, water. So wherever salt goes, water follows. Remember this rule in biology. This is the same reason we do salt water gargles when we have our tonsillitis, when there is any kind of swelling in the throat or infection in the throat. We do salt water gargles because salt. So swelling is because of this water, right? Water accumulates in tissues and cells. That's why there is swelling, right? Or due to infection when bacteria produces this fluid, correct? You understand? So if when we do hot, warm water, salt water gargles, it helps us to get rid of the swelling because the water comes out. Okay. Also, potassium gets reabsorbed. And while these things are getting reabsorbed, along with some nutrients also, so all these things which I have marked in red gets reabsorbed. Write down. Things that get reabsorbed from the PCT includes bicarbonate ion, sodium chloride, water, nutrients, and potassium. But if the PCT is taking so much out, like from the filtrate, to maintain the ionic balance of the body fluids, you cannot just keep negative. So potassium is positive ion, bicarbonate is a negative ion, NaCl also has positive and negative component. Water is, you are taking a lot of solute. You cannot keep, so the electrolyte balance will, the ionic balance will be disrupted. So to, when they take potassium, it has to put hydrogen back in and ammonia. Ammonia anyways, you can just, if you have ammonia anywhere in your cells in the body, which we have, all the ammonia does not get converted into urea. So some ammonia which is produced is also secreted back. So that's the secretion part. So I should not use the red color for this as well. Let me use it's purple color. So write down whatever I'm marking here with purple, these are secreted back. Write down. To maintain the ionic balance, the PCT selectively secretes. The PCT selectively secretes hydrogen ions and ammonia into the filtrate. Hydrogen ions and ammonia into the filtrate. Make sense, everyone? Great. So this is the PCT part of urine, how this filtrate. So we are done with the 80%, okay? But still 20% of important thing, 20% of things are still there. Even if we excrete that 20%, so you can just imagine 20% of 180 liters per day is 10% is 18 liters. So 20% is around 35-36 liters. We are also not capable of losing 36 liters water every day or 36 liter urine every day, right? So which means more reabsorption needs to happen. Okay. Is it clear? Okay. So PCT is done. So the rest 20%, which the PCT could not, negotiate and take back, goes to the loop of Henle. Now, loop of Henle has two components: the descending loop of Henle and the ascending loop of Henle. Now, these two loops, just like it is shown here, you see, pay attention to this. The descending loop of Henle is thinner or narrower, and the ascending loop of Henle, the half part, is wider. It is for a reason. This kind of structure helps a lot. So write down loop of Henle. So the idea is this descending loop of Henle is permeable to water. Write down. So water can go out from it, which means you can still save, the body can still conserve more water from that 20%, but it is impermeable to salts or ions. So write down. The descending loop of Henle is permeable to water, but is impermeable to electrolytes. Okay. But if it is permeable to water and you start losing water, what will happen to this filtrate as it goes down? When it will go the lowest at this point, where the descending loop of Henle ends and the ascending starts, there will be very less water in that filtrate and lots of salt in the filtrate, right? Electrolyte will be very rich. Do you understand that part, everyone? It will be very concentrated filtrate with lots of salt in it.
Yes.
And this is what this structure wanted to achieve, to increase the concentration of this salt, so that what happens is, in the ascending loop of Henle, you can, by simple gradient movement, also conserve or reabsorb salt. So salt starts increasing in concentration as the filtrate goes down and down and down into the medulla. Lot of water is reabsorbed, so the electrolytes gets more and more concentrated. In the ascending loop of Henle, it is write down, ascending loop of Henle has minimum reabsorption, but and is impermeable to water. So the ascending loop of Henle has minimum reabsorption and is impermeable to water, but allows movement of electrolytes both actively and passively. And the simple reason is the very high concentration of electrolyte that was reached in the loop. So it allows the transport or the movement of electrolytes actively or passively through it. Okay. So again, the idea is you removed water in the descending loop of Henle, as a result of which salt will be removed from the filtrate in the ascending loop. And again, the filtrate will be diluted and also less in amount. So you will save more water and more electrolyte from that filtrate. And while doing this, the whole idea of urine formation is to secrete nitrogenous waste. So the kidney cells or the nearby cells will end up dumping more urea into the filtrate, because see, you can see here urea being dumped into this because NaCl is coming out, so the solution is getting diluted, so you have more scope to put in more solute, so urea act as that. Is that clear, everyone? Okay. Now let's come to, we will talk about loop of Henle more because I have to teach you the counter current mechanism. Now let's come to DCT, write down distal convoluted tubule. Distal convoluted tubule. Now, as you see, by the time filtrate reach this point, 80% is reabsorbed back here, water is reabsorbed back majorly in the descending loop, salt is reabsorbed back majorly in the ascending loop, because these are the things, potassium, bicarbonate, salt, nutrients, water, these are the things we cannot lose a lot in urine. Okay, because these are important things. So by the time filtrate come here, it is almost very concentrated. Okay, but still it has some useful things in it. For example, some more NaCl, water, and bicarbonate ion. Okay, so write down in the distal convoluted tubule, a conditional reabsorption. So it is called conditional reabsorption because it is condition based. If you need more, for example, you are on a very good diet, you are drinking good electrolyte based fluid, nimbu pani you are drinking, you are having good food, in that food there is salt, there is water. So if something reaches here, then you can decide not to reabsorb more and you're like, okay, let it go, I have a lot in the body. But suppose you are stuck in a dry area, you went to a wildlife safari on a desert or desert safari, got stuck, now you are thirsty, you're not getting water, you are sweating heavily. In these conditions, the body will suddenly decide, okay, we need to conserve each and every drop of water and salt that we can. So in those conditions, that's why it's called conditional absorption. conditional reabsorption. Sorry. So in those conditions, the DCT, distal convoluted tubule, will start reabsorbing water. Again, I'll mark with red, salt and bicarbonate, but at a cost. Now again, whenever you reabsorb something or you secrete something, you have to maintain a balance. And to maintain an ionic balance, if you are reabsorbing something, you have to also secrete back something into the filtrate, right? So in DCT, a conditional secretion of potassium and hydrogen also happens. So this potassium was conserved here. PCT reabsorbs potassium, but when it comes to a point where you have to make a choice between survival or then you are reabsorbing NaCl, you have to give back potassium sometimes to maintain the ionic balance. Okay, so write down. Conditional reabsorption of sodium and water takes place in DCT. And also bicarbonate ion. And also bicarbonate ion. And selective secretion of potassium, ammonia and hydrogen. There's one thing that NCERT forgot to write here is ammonia, NH3. So NH3 is also secreted back, both at PCT and at DCT. So just mention this part. I think they forgot to mention it in the book. So selective secretion of potassium ions, ammonia and hydrogen ions is done. And why is it done? Because it is important to maintain sodium potassium balance in the blood. So if you are secreting potassium, if you are reabsorbing sodium, you have to secrete potassium. If you are reabsorbing potassium, you have to secrete sodium. Is that clear, Hiram and Janya? Great. So by the time we come to DCT, the urine is already like it's all ready to be sent out through urethra. So it's very concentrated urine which we need. So from here, things go into this collecting duct. Okay. Now, from in one collecting duct, multiple, as I told you, multiple nephrons, give their filtrates, the final concentrated filtrate. And in the collecting duct also, again, if depending on the condition, if a human is fasting or starving or is very thirsty, then some amount of water can still be taken out. Okay, but the problem is if you take out water from collecting duct, if you try to save more water, then some urea also leaks out. Correct? Do you see these lines? So urea also leaks out if water leaks out. So as I told you, there is always a limit, of urea that you can dissolve in water. Urea is still a medium toxic waste. It's not uric acid that you can take more water out without taking the uric acid. So urea also comes out. So this is a decision that body has to take depending on what state or condition of hydration it is in. But if this urea which comes out can again be pumped back into the ascending loop, so no worries. Okay, we have a mechanism for that. So sometimes you see that human urine is very transparent, like watery, almost like looks like water. So just looking at the color of the urine, you can say what is the hydration condition of the person. So when we are dehydrated, we are not drinking water properly. The urine which is excreted out turns yellowish, more and more yellow, correct? Yes, no? You understand the correlation? That's because the body is deciding to conserve as much water as it can, but it has a benefit that you get water, so it will absorb water, reabsorb water from everywhere, ECT, loop of Henle, DCT, and also from collecting duct. So the benefit is you conserve water, but the loss is that you also cannot then excrete more urea. So dehydrated body also accumulates toxic urea over time. So that's why you should be properly hydrated. That proper amount of urea can be dissolved in proper amount of water and can be thrown out of the body in the form of colorless watery urine, which is a healthy urine. Okay, so does this diagram make sense, everyone? Yeah, so write down about collecting duct. Finally, write down final thing is collecting duct. PCT, loop of Henle, DCT is done. Collecting duct, write down. Collecting duct extends from cortex to medulla. So it's a very long duct, cortex to medulla. And if required, large amount of water can be reabsorbed. If required, large amounts of water can be reabsorbed from collecting duct. Okay, to produce a concentrated urine. To produce a concentrated urine. Okay, and write down, while doing so, while you are reabsorbing water, while doing so, the collecting duct also allows, while doing so, the collecting duct also allows some urea to pass through, which means it's just allowing some bad stuff also to go back to the body to maintain the osmolarity, osmotic balance. It's very important. It's always a trade-off. You cannot get all the bounties and cannot throw all the bad stuffs. It's a balance. Okay. And write down, one thing that they have not shown here is that even when it is throwing water out, it can also, collecting duct can also allow selective secretion of hydrogen and potassium ions to maintain ionic balance. So write down, collecting duct also plays a vital role. Collective duct also plays a vital role in maintaining the pH and ionic balance of the body. Oh, sorry, of the blood. Maintaining the pH and ionic balance of the blood by selective secretion of hydrogen ions, which is H plus, and potassium ions, which is K plus. Is that understood, everyone? Great. Very well. Okay, so this is a very major technical thing in this chapter with lots of concepts. So I'll stop here for a couple of minutes. A break of 2 minutes, 2 to 3 minutes. I'll fetch some water for me. Please look at this diagram, look at every part of it, every labeling of it, and see if you do not understand anything. And let me know, okay? So a quick break for a couple of minutes here. Okay, Hiram and Jani. Okay. Okay, so are you there, people? Come back. Hiram, Jania. Am I audible? Hello. Yes. Okay. So Hiram is there. Jania, are you there? Just confirm your presence. Any doubts, Hiram? Is this diagram completely clear to you? And all the parts that of the nephron that participate in urine formation is clear? Okay, 100%. Great. Jania, we are still waiting for your confirmation if you are there. And if you have any doubt. Okay, no doubt. Great. Now, just remember that whatever we studied till now, just for sake of simplicity, it is just the nephron. But in reality, it looks something like this. As I told you, it's not just the nephron, the PCT. So to show you there, the PCT was opened, extended, then it was loop of Henle descending, ascending, and then the DCT was extended. There was no blood vessel around it, right? Around this diagram. So this is not the complete story yet. This is the part of the story. Now, to get to know the complete story, I have to tell you about something which is known as the counter current mechanism. So this is what I'm going to teach you. Write down the the counter current mechanism. Now, what do we mean by counter current? Counter means to face, right? To oppose something. Now, one thing so whatever is being reabsorbed, this HCO3, NaCl, water, nutrient, potassium, water, salt, where is it going? What do you think? Where is it going? It's going back to the blood, right? Because that's where we took it from. That's where the Bowman's capsule took it from. So whatever is reabsorbed goes back to the blood. For that exact reason, the blood vessels do not leave, the efferent arteriole do not leave the nephron after the glomerulus part. It stays with the PCT, DCT and loop of Henle in a network manner because it has to get back a lot of things. So this counter current mechanism, write down, it is also called the mechanism of concentration of the filtrate. The mechanism of concentration of the filtrate. Okay, and let me insert the image for you. NCRT. Yes, can you see this image, both of you?
Yes.
Yeah, this is the image that you have to understand. There are two things in this image. Yes, now, in this image, pay attention that still two things, but two are different. This half part is the nephron, about which we have already studied, and this part is the arteriole. Okay, the vasa recta, the blood vessel in which everything goes back. Okay, that's why there is this. And if you see the direction, pay attention to the direction. The direction in which things move in the nephron is this. So from Bowman's capsule, it comes, goes into the Bowman's capsule. Follow the arrow. It moves like this into the loop of Henle. This is called descending because it goes down, and it goes up. Ascending, goes into the DCT, and then finally goes into the collecting duct. Now just superimpose this picture on this because they both are in a network, right? And we will realize exactly when direction is downwards in a nephron, the direction is upwards in a blood vessel. Correct? Do you appreciate this, people? And when the direction is upwards, the direction is downwards in a blood vessel. Yes? Now, if I keep, so this loop, sorry, this part interacts with this part, and this part interacts with this part. Okay, I use both blue, so let me not use blue. Use a green. So this green part interacts with this green part. Make sense, everyone? And through this movement, what happens is, now just use the knowledge that we saw here, things are getting reabsorbed. So when things gets reabsorbed and there is more water and less solute, the value, the what do you say, this osmolarity value decreases. It is 300 around at the PCT. As it goes down in the loop of Henle, so this is loop of Henle. In the loop of Henle, what is coming out? Water, right? It is permeable to water but impermeable to salts. So water is coming out, but salt cannot come out. So the osmolarity keeps on increasing. From 300 in the PCT, it becomes 400, 600, 800, 1000, and it is maximum at the bottom where you have lost water, but all the salt is concentrated. Make sense, Iram and Jani? Yes, no? Am I audible, people? Make sense? So osmolarity is increasing because the salt concentration is increasing as the filtrate goes down the loop of Henle. Okay. Then again, as it goes up, salt starts coming out. You can see all of this salt is coming out. And again, the osmolarity will decrease 600, 4, 3 and come back to 200 before it is thrown into the collecting duct. And at the same time, if you see in the blood, what's happening? When osmoregularity is decreasing in loop of Henle, it is actually increasing in the blood because whatever the salt which is coming out is going in the blood. That's what is called the reabsorption of the salt. So from 300 here, this was 600 at the bottom and 300 at the top. The blood is 300 at the top and comes down to 600 at the bottom. It absorbs, and it also reaches maximum at the down. Then again it decreases because this water which is being reabsorbed comes back into the blood and NaCl goes out. So this is called balance, the osmolarity balance between blood and the nephrons. And this is how we decide that how much electrolyte balance should be in the blood. Whenever it increases or decreases, this activity of nephron, so that's modulates it. So that's called a counter current mechanism, where both nephron and blood vessels counteract on each other's osmoregularity. Now you can write down the counter current mechanism helps. Write down the counter current mechanism helps. We will talk about it in some more detail in the next class, where we will finish this chapter, because after this it's all theory. So I'll give you a homework to go and read this chapter, the theoretical part, which involves regulation. So you will read about some hormones. One hormone you have already read in the endocrine chapter, which is ANF, atrial natriuretic factor. Remember that was helping the kidneys to maintain the blood pressure, correct? Yes or no? Can you connect to knowledge? Am I audible, people?
Yes.
Yeah, okay. So write down this last point, that countercurrent mechanism helps to maintain a concentration gradient. A concentration gradient in the medullary interstitial tissues. In the medullary interstitial tissues, which helps in movement of water from nephron to blood and blood to nephron. Or sorry, from nephron to blood basically. It doesn't go back from blood to nephron. From nephron to blood, therefore concentrating the urine. Therefore concentrating the urine. Okay. So I will stop here. Let's stop here for today and take the rest of the parts in next class. Okay, where we'll finish this chapter. Any doubts, Jania, Anila?